Hi everyone, today I'm delighted to have Marc Voigt, CEO of Immutep, a roughly $400 million market cap company listed on the ASX. And this is going to be a really interesting episode because Immutep is targeting some horrible diseases and they're doing their absolute best to cure them. So I'm looking forward to learning more. So Marc, welcome.
Yeah, Michael, thank you for having me.
Thank you. Thank you for coming. So I thought we'd start with lung cancer. So can you give us a bit of background around how it presents? How old are people? How common is it? Just a bit of background around the disease itself.
Unfortunately, lung cancer is very common. It's a leading cause of cancer deaths globally, with around about 2 million cases of people dying. It's a disease which, unfortunately, you will not discover easily. So very often it's diagnosed already at a later stage, which makes it so hard then to treat, because diagnosis is a very important piece to fight the disease. So we have here a high unmet medical need because with best available treatments, still the median so-called overall survival, so the time usually a patient lives, is about 2 years when it comes to the metastasized setting.
Right. And how does it present? You say it's very later stage. Are people quite ill at that point?
Not yet. Very often earlier on you actually don't recognize it. And this is exactly the danger. So if you don't go, for instance, as a smoker to regular checkups, you may not discover it until you have already then some symptoms like a longer coughing, eventually maybe some blood. And then it might be too late to have surgery applying to remove the primary tumor because it already metastasized, meaning that in any case you need to have additional treatments.
Right. So really you want to find it before it's metastasized, but generally that's asymptomatic at that point.
Correct.
Right. Is this one of those cancers where it'd be helpful to have screening, or is that too hard or impractical?
No, there are efforts to do that, but the day-to-day reality is that you have most cases metastasizing and have it at a later stage, so-called stage 3, stage 4. This is unfortunately the reality. You also have to weigh the burden in terms of diagnosis. You need to have the discipline from the population. It needs to be affordable at the end of the day, and you have to weigh all these different factors. So it's known since decades that the stage of diagnosis and then the treatment options, the earlier the better. Reality is still that it's very often too late and that you have been reaching metastasized stage, or even if you diagnose it early, that it still comes to a relapse or metastasis later on.
Right. And how old are patients usually when they get it? And are they mostly smokers?
Mostly smokers, but it's not the only risk factor. There are many different other risk factors like genetics, radon, so exposure to asbestos, so many different other risk factors. And patients are typically a bit older, but you can also get lung cancer if you're younger. It's just the likelihood, which is increasing over time.
Right.
Like for most cancers, actually.
Yeah, got it. And then at that stage, once you've been diagnosed, what's a typical patient journey?
First of all, you can hopefully have still surgery so that your primary tumor is being removed. Radiotherapy is an option. You have chemotherapy, you have immunotherapy, targeted therapy. So there are different interventions you can take at an appropriate stage.
Right. So my understanding is that chemotherapy was one of the traditional treatments. What were the typical agents that were used?
Like 5-FU, the platinum salts, so different chemotherapy families and recyclins, so different chemos really. And it also depends which type of lung cancer are you having, small cell lung cancer, non-small cell lung cancer, which accounts for around about 85% of the cases. And at which stage you are, if you look at non-small cell lung cancer, actually also if it's squamous or so-called non-squamous. So there are different chemotherapies and different other treatment options.
So what's the difference between small cell and non-small cell?
The type of cells are different, obviously larger or smaller cells. And then you still have, for non-small cells, squamous, non-squamous, so where you have different cell types which are differentiated and which require different treatment and intervention options. I mean, cancer is a differentiated disease, obviously. You have the primary tumor, you have then the metastasize, you have the primary tumor metastasizing, for instance, into the liver, into the brain. So different organ systems are being affected.
Right. Why don't we talk about immunotherapy then, because that's getting us closer to Immutep.
Sounds good.
So there was a bit of a revolution in that, and it seems, immunotherapy was extremely effective for a small number of patients, perhaps 20 to 40% across solid tumours, notably melanoma, which is obviously very common in Australia, and lung cancer. So you're able to describe the immunotherapy, how that works, and how it works in lung cancer?
Yeah, it's actually a longer journey. So this revolution, and you're right to talk about a revolution there, it took actually decades. Some decades ago, it was believed to be a ridiculous idea that your own immune system can kill cancer. So no one believed in that. And it took the clinical oncologists and the scientists, the researchers beforehand to come up with some treatment options like anti-PD-1 therapy. So cancer immunotherapy is predominantly associated with the so-called immune checkpoint inhibitors, where you keep the T cells active. T cells you can refer to as soldiers in your immune system, which are doing the killing job. And to keep those soldiers active, you block immune checkpoints like PD-1, CTLA-4, LAG-3, little triggers in your immune system with which you can up or down modulate the immune system, and then actively fight cancer. And about 10 years ago, 2014, Keytruda has been approved. Before that, another drug, anti-CTLA-4 called Yervoy from Bristol Myers Squibb. So this revolution is going on since more than a decade. But indeed, it has been leading to a shift in terms of existing standard of care, day-to-day treatment of patients. And in terms of lung cancer, 7 to 8 out of 10 patients are being treated with cancer immunotherapy, immuno-oncology, such as Keytruda, the most powerful drug. But still, to your point, the majority of patients does not benefit from those treatment options. Still, if you have 20, 30, 40% of patients responding, that's a huge win.
Absolutely. It's crazy to think that was only 10 years ago.
It is, but this journey, as previously mentioned, is going on for a longer period of time. These immune checkpoints have been identified in the 80s and early 90s. And it takes a while until those revolutions take place. So it's not coming overnight. Very often innovation is not recognized early on, and then you have a breakthrough. And it's also very interesting to look at the clinical development. So it was not a pre-planned success, but it was a breakthrough innovation where simply one saw in the clinical trials, here are some remarkable results. Let's aggressively roll out the case and see a big success.
So 70 to 80% of patients are treated with Keytruda, but only works in a small fraction of those. How well do we understand why it works in some patients and why it doesn't in others?
That's a good question. We and the agencies and researchers are looking into so-called biomarkers, so where you assess the biological setup of a patient, of the individual, in order to see if therapy has a chance to work. For Keytruda in non-small cell lung cancer, it's the so-called TPS score, so where you measure a PD-L1, and the higher this score is, the better the patient is supposed to react to anti-PD-1 therapy, such as Keytruda or Opdivo from BMS. And round about a third of the patients have a so-called high TPS score, and 2/3, unfortunately, can only expect a suboptimal or no response. And even in the TPS high patients, of course, you do not have a 100% response rate, but maybe 40 or 50%. And this is why you typically go into combination treatments, like with chemotherapy, or combination of two immunotherapy options like anti-CTLA-4, anti-TIGIT, anti-PD-1 combined. And this is also what we do.
Right. So that might be a good point to move to Immutep. So what's your approach and how does it work?
Yeah, we have, I believe, a very, very good approach because we are off the beaten path of the T cell. Previously, I mentioned immune checkpoints on the T cell. You keep the T cells active, the soldiers of the immune system, let's call them. We focus on the generals of the immune system, the so-called antigen-presenting cells. These can be dendritic cells, for instance. Those cells are responsible to provide the soldiers with the information whom to attack. Because obviously the immune surveillance failed, otherwise we wouldn't have cancer, as the T cells are not able to recognize cancer. So we provide that information, we boost that interaction as one effect. Second effect is we help to draft more soldiers. And this is pretty powerful, and we can actually measure that in patients. So we activate the immune system differently and deeper than some of the traditional approaches. And we have been testing this preclinically and then finally clinically with, I believe, very remarkable results.
Right. And how does it actually work? What are you actually targeting?
We are targeting MHC class 2. That's a receptor on the antigen-presenting cells. So a dendritic cell, an immature dendritic cell, has a specific subset of MHC class 2. I will not go too much into the specifics, but it's those expressed in lipid rafts. We created a fusion protein, which consists of the 4 extracellular domains of LAG3 fused to human IgG1, so that it's not recognized as a foreign protein by our immune surveillance, so that it can work. This docks an MHC class 2, that's a major ligand to LAG3, and then sends an activation signal. So it's an agonist, an activation signal to the dendritic cells. You can see that nicely in vitro with the dendrites popping out after a few minutes. And then you see the proliferation of CD8 T cells, CD4 cells, natural killer cells. So it's a very broad and deep immune activation in the crosstalk of innate and adaptive immune system. And this works very nicely with immune checkpoint inhibitors, as we have been showing clinically, as those are keeping the T cells, the soldiers, active. So we create more T cells. We create and activate different other immune cells. We provide the information whom to attack or boost that interaction. And Keytruda, for instance, keeps those soldiers active.
Right. So how specific is it to cell type in terms of, or is this a broad immune?
Interestingly, we don't treat cancer. We treat the patient's immune system to actively fight cancer. So what you need to have are antigen-presenting cells. If you, and maybe 3% of the patients don't have cells called monocytes. If you have monocytopenia, there might be limitation for the treatment. The immune system itself is very complex. So will it work everywhere and in every single patient? Certainly not. But in principle, it's activating the immune system of a patient to actively help the body and the immune system to fight cancer.
Right. So it's interesting. So it's really targeting the immune system.
Yes.
Understood. So how long have you been at the company?
I've been with the company now since about 12 years, about 10 years as CEO.
Right. And what data got you so excited at that early stage?
First of all, the concept, and then the revolution taking place in terms of immuno-oncology, Keytruda being so successful, so great data, the chance to help patients, and to see this medical success. And then, of course, it's about being innovative. And this is what we have been doing in terms of clinical research. We started phase 1, phase 2. Now we are at phase 3, so at a mature stage, hopefully the final step before registration.
Right. So why don't you take us through the lung cancer data then? Because you have phase 2 and there was a trial result released recently as well, which is hopefully some kind of read-through into your big phase 3.
Indeed. I mean, we release data along the years. Actually, we have been presenting at top scientific conferences such as ESCO in the United States and Chicago. There are typically 40,000 oncologists, industry representatives. We have been the first Australian company ever presenting at a late-breaking oral at SITC in Boston. It's a special immuno-oncology or cancer immunotherapy conference. And we have also been presenting at ESMO. That's a European version of ESCO. So we have been presenting at top conferences. We had the honor in the one or the other conference to be the first Australian company to do that. And the data we saw has been very remarkable. From a study called TACTI-002, which we are running together with Merck from the United States, Keytruda plus eftilagimod, we saw very good median overall survival. And that's the gold standard. For the patient, it's of course most important to live as long as they can, and this with a good quality of life. So we saw for patients, for instance, a median overall survival of more than 35 months. And you can benchmark and compare that with historic trial results of 16, 18, maybe 20 or 22 months median overall survival. So it's a big win. And it was so exciting that also the Nasdaq bell tower in Times Square in New York has been lit up and congratulated us on the lung cancer data. So that has been very exciting. That was in 22-23. And you have been referring to new data This is from another clinical trial called Insight 003, looking at eftilagimod plus Keytruda and chemotherapy. And there we have been press releasing recently very exciting results with 32.8 months median overall survival, good progression-free survival, good overall response rate.
And is this, it's probably not placebo-controlled, but controlled versus standard of care, or is it just open label?
Versus historic control. Typically in phase 1, phase 2, you don't go into randomized trials. Sometimes you do that, but typically you compare that to the large trials which has been leading to approval for the pharmaceutical industry.
Yeah, I guess the challenge with these things is then how do you control for, all the different things that could come into whether a patient is going to react or not react to the treatment?
Yeah, how can you actually, let's make the question even a bit harder, how can you rely on the data that you're not seeing a false positive or false negative results? This you can achieve via a good trial design. For instance, TACTI-002 is a so-called Simmons two-stage design.
That's your phase 2 trial.
That's a phase 2 trial, 114 patients. So first of all, the trial size is in a way that you do not have, 10, 15 patients, and there you see by chance very good results, maybe from a single center. So we have been conducting or are conducting this trial in 6 different nations, including Australia, the UK, United States, in different trial sites. And we implemented with our collaboration partner Merck different hurdles, meaning we said before we started the trial, we want to at least see 35% overall response rate. And then we recruited the patients. You ensure that you do not cherry pick patients, but you take the normal proportion of patients you would typically expect in larger settings. The data is published. Obviously, we have been seeing that. And by the way, a little bit less patients who would typically react positive to Keytruda. You would normally expect. So the chances were stacked against us. And I believe...
Could you just explain that a bit better?
So you have, I previously mentioned the so-called TPS score.
Correct.
High TPS score means patients will react very likely, very well to Keytruda. 1 to 49 would be suboptimal, and below 1, patients would typically not react to Keytruda. These biomarker-associated patient grouping. We had less than around about 22% of patients having had TPS high. So only a smaller proportion than you would normally historically expect had a good chance to react to Keytruda. Meaning a larger proportion of patients had typically worse prognosis. So we had more patients which were more difficult to treat with Keytruda. So we treated them with a combination, and we saw overall the remarkable results in terms of overall survival, overall response rate, duration of response, progression-free survival. So a consistently positive picture across all different nations. So it was not an outlier that one center, for instance, has been recruiting, let's say, half of the patient, and by chance those patients have been doing well, but the others not. And we have been doing this together with Merck. And by definition, you are under a lot of scrutiny, and they have also been doing the biomarker testing. So that was under their control. And we prefer that setting, actually, because you don't want to fool yourself. You want to go after medical innovation with good, robust, and solid research. And it could always happen that you identify a setting where your drug doesn't work. That's not a pity. That's simply the consequence of medical research. And we saw very good results in non-small cell lung cancer in this trial. We saw it in Insight 003, as per last week, very exciting results. And we are hopefully going to see it in TACTI-004, our lung cancer clinical trial, the phase 3 registration trial.
Yeah, love to come back to that. But I was just looking at in the Insight 003, so this was the one that was released November 14. It's November 19 now, so it's only a few days ago. So it says mature data in patients, minimum follow-up of 22 months shows excellent results, well above historical controls. But the question is, these are the patients that have had the minimum follow-up of 22 months. Does that mean they're the ones that had already reacted and then they're more likely to perform well? It then follows up to say data from all patients demonstrates significant improvement of overall response rate. So the question is, was there like a bias there? Like, how do you interpret that as somebody who's just looking at this data fresh?
No bias. It's just that you have a number of patients who are under treatment for a longer period of time. When you recruit a clinical trial, some patients will come at day one or after 1 month, 3 months, 6 months. So you have a continuous patient recruitment into a trial. Meaning some patients have experienced the drug for a longer period of time, and therefore you can measure already overall survival.
So if somebody sadly passed away before 22 months, they're still in that data. That's kind of what I'm
asking.
Yeah,
definitely. You cannot.
So it hasn't been poisoned.
Yes.
Right.
You, of course, need to include the patients who have been then passing away under treatment and had the follow-up visit, of course, yes.
Right.
Otherwise, you cannot come to the median. And we provided the data with, for instance, minimum follow-up of 22 months, showing to the market and indicating that it's mature data. So it's not after 6 months, everyone is still alive, hooray, but that you have a long minimum follow-up and that you look at mature data.
Right. So it's not just the subset of people that have already survived 22 months.
No.
No. Right.
It's all the patients. And of course, we will recruit eventually, or the investigator, it's an investigator-initiated trial, up to 50 patients, and then more data to come. So it's when you detect, when some data is mature, it's going to be released, and then, of course, continuously until the study finishes.
Yeah, so you had a phase 2, you have this INSIGHT trial, but really it's the big phase 3. What was the reason for doing this particular trial? Because it's smaller, it's open
safety,
label.
Predominantly, and first of all, safety, because we wanted to look at the safety of the combination of FD plus Keytruda plus chemotherapy. It's a combination at the end of the day of 3 different types of therapies. And also, of course, as this is the predominant standard of care, so this is applied most in the day-to-day clinical care, if it works in that setting. And this is what we did.
So let's go into the phase 3. How's that designed? And what are the 2 arms? Is it head-to-head? How have you constructed that trial?
Yes. So the phase 3 clinical trial, we call it TACTI-004. It has also a keynote designation because we teamed up with Merck. Actually, the first time since more than 2.5 years that Merck teamed up with a company in a clinical collaboration setting to conduct a phase 3.
Right. What is that relationship? How does that work in this trial? Are they committing funding? Are they committing drug?
They commit roundabout commercial value, 120 million US dollars worth of drug, jointly design the clinical trial, and practically they put their reputation on the line because this is a clinical trial which is aiming to set an improved standard of care for the most important cancer indication out there. So the total addressable market is round about 24 billion US dollars. And it's important to understand that 35 to 40%, give or take, of the revenues from Keytruda, and Keytruda is a top-selling drug in the industry across all indications, not only oncology, but across all indications, are being generated in lung cancer. So it's a sweet spot, the commercial sweet spot, the medical sweet spot of Keytruda. And the collaboration is, of course, an agreement, many different pages where you define the interaction between the parties. And, of course, this is under a lot of scrutiny, especially when you do it for the first time since a few years. And a big step, and for me, a big commitment from the other side. So a phase 3 is a big clinical trial. It's 750 patients in this case. So it's a big undertaking. Final step, if successful, before registration. And I believe it's very pleasing for us that we do this together with one of the key players in the industry, together with the top-selling brand of the industry. So that's a very strong value proposition.
Do you know when
based on data.
Do you know when Keytruda's patent runs out?
The so-called patent cliff starts to kick in by 2028.
28, so that's only a few years away.
Few years away. It's tomorrow by the standards of our industry.
And if everything went well, potentially this could help them expand that somewhat. In combination.
Indeed, we have patent protection on the combination, for instance. We have different other patents. So this could be one of the very viable options to prolong exclusivity for Keytruda.
Right, got it. And in that trial, what's the timeline? What can we expect?
So the trial will start very soon. We provided earlier guidance in terms of end of this year, Q1 next year. And
so Q1 next year.
Or end of this
first
year.
half?
So study start is on the one hand side the regulatory approval, and we will certainly aim to have this study in more than 20 countries and more than 150 hospitals around the globe, including Australia. And then, of course, a recruitment starting. So this is imminent and within our guidance. We are working very hard on that. It's a lot of work. And then we are going to recruit, and we will have, it's event driven, so it depends, of course, on the patients being recruited, but a futility analysis in a good year from now, maybe early 26, give or take, and the chance to see the interim analysis by back end of 26 on which we can file. And we have a so-called dual primary endpoint, progression-free survival and overall survival. We and our collaboration partner have been investing a lot of effort in terms of the statistical grid, in terms of the trial as such. It's important to understand that this clinical trial is not a small randomized trial, so where you need to have, by definition, normal statistics apply, small numbers, you need to have huge differences in order to detect statistical significance. This is a trial which is aiming at a reasonable improvement versus standard of care. So a control trial, you have been asking about the two arms. On the one hand side, eftilagimod plus Keytruda plus chemotherapy versus placebo plus Keytruda and chemotherapy. So it's a blinded, controlled clinical trial. Don't go too much into the specifics, but also stratified for a number of factors. So where you should or you want to ensure an evenly distributed patient population in the two arms, and then you're going to see the readouts.
So in that futility analysis, what as investors can we read from that when it comes in about a year?
Or it should be another big boost of confidence, if positive, of course, because it will further de-risk the trial. So you do know that based on the first analysis, the trial is not futile, continues to be recruited, and should be a very good boost of confidence for that clinical trial.
So will that suggest it's working to some degree, or is it just that it's not working?
No, that it's working to some degree. So there's, of course, a hurdle you need to meet. It's based on early efficacy parameters. So this, for instance, overall response rate. There's an independent data monitoring committee, so very high quality standards. It's not the company looking into that and then deciding, hey, look, it's good enough. It's an independent committee looking at that, assessing the data, go, no go.
Got it. And then the actual readout would be about a year later?
Yeah.
About a year later. They're long journeys, aren't they?
Or not too much. It depends from your perspective.
I mean, from the start. I mean,
oh yes,
this is the
from the start it is. I mean,
multiple trials.
True. Medical research, of course, takes time. So the revolution we talked about is not coming overnight, and especially when you want to see some overall survival, it of course takes some time.
Right. Why don't we talk about commercials? So what do you think the commercial opportunity is for this drug if it's approved?
The commercial opportunity is simply very huge, and that's not a marketing statement. Everyone would like to work in huge markets. It's simply met by reality. Total addressable market, non-small cell lung cancer first line now is around about 24 billion. It's supposed to double by 2031.
Why is it doubling? Is that?
Because you have more patients, aging populations, and you have simply the increase of the sales prices. These are the predominant driving factors for that. Right. That's all based on market research. And we do not need to create the market. That's also important. We walk directly into a multi-billion-dollar sales opportunity because Keytruda is there, patients are being treated, and they would get one additional shot. Eftilagimod, it's delivered subcutaneously, so an easy route of administration. You press a syringe, that's practically it. And then you have potentially a very good benefit. So the commercial opportunity from a sales perspective is very big. However, for biotech, very often it does not come to own sales because companies are being acquired, licensing deals being done, and this often also happens prior to final data.
Right. Do you have some feel from what recent acquisitions in the space have been done at?
Yeah, if you look at the stats, the sentiment in the past few years from the big pharmaceutical industry has been changing. In some prior years, there were 50% of deals done at preclinical phase 1, phase 2 stage, and about half of them at phase 3 and commercial stage. According to latest research, more than 80% of transactions, and I talk about takeover transactions more than $1 billion, are being done at phase 3 and commercial, and only less than 20% actually are being done at preclinical phase 1, phase 2 stage. So you see a shift of the pharmaceutical industry towards a risk reduction. The more data you have, the better it is. They are paying higher prices. And you see, of course, very, very exciting transactions in the past. Every deal is unique, but you need to deliver the data. And the deals and transactions as such in the biotech space, I believe it's publicly available information that 90% of transactions are being done with one bidder only. So a good dream of whether you sell a car, a drug, a company, of a competitive situation, an auction situation, is only happening in the minority of cases. So you should take good care as a company that you know multiple different players, that you have the one or the other person at speed dial. So if the opportunity presents itself, that you can also create the right competitive tension.
Right. And who do you think would make a natural acquire? Obviously, there's a relationship with Merck.
We have a very good relationship with US Merck. We have also a very good relationship with German Merck, by the way. We have 2 other clinical trial collaborations there, originally with Pfizer and German Merck, and then German Merck acquired the rights to a drug called Avelumab. And there was a study running in urothelial cancer. We have also other relationships, which are covered by CDA. So there's definitely interest in eftilagimod. US Merck, we have a long-standing relationship. We have a phase 3 collaboration, but we retained also all commercial rights, meaning we didn't kill the competition, if you like.
Right. Why don't we move on to head and neck cancer?
Sounds good.
What are you up to in that space?
Head and neck cancer, we have a clinical trial in first-line head and neck cancer. We had also a successful one in second-line head and neck cancer. And there we saw also some data in different sets of patients. And moving forward, we will predominantly focus on the patients with a low so-called CPS score, same story like with TPS. So CPS below 1, meaning where Keytruda is not approved actually. And where we saw very remarkable results. We saw a 7-fold increase of the normally expected response rate. And actually prior to Christmas, we are going to see some new data. Right. So that's potentially also very exciting. Let's see.
What's the data? Which trial? Like how many people, which stage?
So this is in CPS below 1. It's 31 patients. Head and neck cancer is a very aggressive disease.
31 patients.
31.
31.
Interestingly, those patients have only approved chemo backbone options. So every approved standard of care has a chemo backbone. But a number of patients, unfortunately, can't tolerate chemotherapy, or they outright refuse to take chemo. And that's a more aggressive regime in head and neck cancer compared to lung cancer, for instance. So we have here a chemo-free setting. It's Keytruda plus eftilagimod. And potentially we could offer those patients and everyone else with currently very strong data, a chemo-free alternative. So that could address one of the highest unmet medical needs in head and neck cancer. Head and neck cancer overall, commercially not as big as lung cancer, of course. If you look there at the market, it's around about $3 billion. About 20% of patients are CPS negative, but could be very nice options to move forward. So let's look at the results first, the new results. And then we certainly will discuss also with the FDA regarding the right path forward.
Right. So what's the relationship with HPV and head and neck cancer?
HPV positive and negative. HPV means it's virus-induced disease. The reality is head and neck cancer is very often associated with alcohol abuse, tobacco abuse, or virus-induced disease. And of course, for the disease or the onset of the disease associated, let's say, with alcohol abuse, very often patients are in not so good shape. Meaning, if you have the disease caused by a virus, your chances are often better because patients are often simply in better shape to live longer than regarding HPV so-called negative patients. So it has to do with the baseline characteristics, so the demographical factors, the biology factors of the patients. And there are a number of different factors, like the debate around male, female. Female are supposed to do better in this disease compared to male. Around the location of the primary tumor, because head and neck cancer is very heterogeneous. You squeeze 4 different cancer types into one. You have cancer could occur in the oral cavity, in the larynx, and it's associated then also with different so-called prognosis. The ECOG plays a role. That's practically the performance status of a patient. Can they still walk? Are they in good shape or not so much? Weight loss and so on and so forth. So it's quite complex.
There's a lot of categories there. So
yes,
a patient
indeed.
in trial with 40 people, it's very hard to control or really. But I guess you have to start small and then go from there. With the data
You have to start small. You can, of course, also start big, but you have to weigh the resources. You have to weigh the commercial opportunity. And you have to see also which risk you are going to take. And you deal here with human beings, with patients. So if you, first of all, why do you do phase 2? You do phase 2 typically in order to detect the patients, a group of patients who can benefit from the treatment so that you have a better targeted phase 3 clinical trial. And there to go big from start, I don't know if that's the right approach.
So in head and neck, where of those categories, which do you think are most promising?
I believe it's CPS negative, not because I believe there is CPS greater equal 1 has been particularly bad or disappointing. I believe there are some very nice sweet spot in that data set as well. If you look closely, you will see that we published results where you see a statistically significant difference in terms of the absolute lymphocyte count, so that you have with eftilagimod and Keytruda more immune cells created compared to Keytruda alone. That was the very first time we were able to show in a controlled setting with Keytruda that we are able to do that. So it's now, if you like, proven that we drive the immune system in a very powerful way. And we have been showing that before, but not in a controlled setting, only versus historic control. So that's very powerful. This is a take-home message for from the randomized part of the study. But there you have also more competition, more resources would be required, and we will certainly not enter into any financial adventures. We will focus on the key value driver, which is non-small cell lung cancer.
Right, got it. In any case, there's more data coming out, so...
In any case, I promise, and actually before Christmas.
Before Christmas. Excellent. Well, good luck with that.
Thank you.
Why don't we move on to your other programs? Why don't you describe what you're up to in autoimmune disease?
Autoimmune disease. So let's switch gears. There would be also more indications, if you allow me that statement, more indications in oncology, like soft tissue sarcoma, breast cancer, urothelial cancer.
And this is all about just stimulating the immune system.
Correct.
In a similar way, using FT.
Correct. Going back to the earlier statement that we treat indeed, exactly as you said, the patient's immune system. And this can work in a variety of different indications. This is why you have a pipeline in a product, if you like, multiple shots on goal. Will it work everywhere? Certainly not. But it has a chance to work in multiple different settings. And this is what you see with Keytruda being approved in well above 30 indications. So we can achieve the same thing. But let's go to autoimmune diseases, as you suggested. Autoimmune diseases is the second field we are working in. It's a very exciting field. Conceptually, in oncology, you would like to have an active immune system. You would like to help the immune system to actively fight cancer. Now, in autoimmune diseases, you would like to down-modulate the immune system because your soldiers, the CD8 T cells, provide friendly fire to your own organ systems, and you would like to bring it under control. Now, what we did is we designed, and this took our Chief Scientific Officer, Frédéric Tribel, who actually discovered LAG-3, worldwide leading scientist, about a decade, to design an antibody which is directed to LAG-3 on the T cell, and is using LAG-3 there as a marker to identify rogue LAG-3 positive T cells, because these are the guys who are attacking the organ systems, if you have psoriasis, for instance, the skin, and are accumulated at the disease site, if you like, the start of the inflammation cascade. So it's a very specific upstream approach. The beauty of the program is that it could potentially address more than 90% of the autoimmune diseases. It's not a symptomatic approach, so where you try to suppress psoriasis, the plaques, the redness and the thickness of the skin, but where you try to go after the root cause of the disease, the chronically LAG-3 activated T cells. And we have been working for years in so-called preclinical development. So we had in vivo studies, toxicology. We are GMP manufacturing the drug, or our engaged contract manufacturing organization is manufacturing it. And since mid of this year, we're actually in clinical development in an exciting phase 1 clinical trial.
Right. So what's the phase 1 clinical trial in exactly? Which condition?
Yes, it is in healthy subjects. That sounds boring, first of all, but it's not. Very often you look in phase 1 clinical trials after safety only. In oncology, you treat patients. A lot of other diseases, if they're not life-threatening, you go after healthy volunteer first. This trial will deliver safety data on the one hand side, but it will also look into the biology, and it will be it is a blinded phase 1 clinical trial, placebo-controlled actually. How do we achieve that? So we teamed up with a world-class research institute in Leiden in the Netherlands called CHDR, and they developed a test where you, in a healthy volunteer, deliberately irritate the skin so that you have a local inflammation, some redness, and then you give either placebo or you give, the name of our drug is IMP761, another weird acronym, but it is what it is, I can't change it. And then you see if the drug is doing what it's supposed to do. So if you see immune suppression. So it's not looking at safety only, but it will tell you the proof of concept and has a biological read-through. And in a phase 2 setting, one would then go after a specific disease, could be type 1 diabetes, could be rheumatoid arthritis, could be psoriasis. So there are a variety of different options. And we see quite a lot of inbound interest for that program as well. It's not only eftilagimod, and IMP761 is not shining into the spotlight so much. We hope that this is going to change. But we will then figure out, not unlikely in conjunction with another party, what to do for phase 2.
Right. That sounds very general then, if it's healthy people that you're irritating their skin. So how are you going to choose between all those different autoimmune diseases?
That's a very challenging question because there are, I believe, around about 200 autoimmune diseases. So you would go after a specific category. Maybe first of all, it's so-called TH1, type 1 diabetes, rheumatoid arthritis. The beauty and the challenge of the program is that it's an upstream approach. So that's not targeted to one specific autoimmune disease, multiple sclerosis or whatever, but you could address a variety of different autoimmune diseases. So you can look, for instance, at psoriasis for mechanistic reasons, because it's easy to measure. It's a simple phase 2. You see on the skin without any difficult biopsy in the body. You can make a punch biopsy simply. You can measure there the effect. Or you go, there has been an experiment by another group in terms of juvenile arthritis. You can go after rheumatoid arthritis. But we have some confidential discussions around choice of indications and what could be a good way to move forward.
Right. What's the current treatment for psoriasis?
You have TNF-alpha, for instance. So you have different, more symptomatic related, so to suppress the local environment, practically symptomatic related treatments.
And this is a small molecule drug, is it? Or
yes.
Is it a peptide?
Yes. You have different small molecules. You have also biologicals. You have also drugs like Lemtrada from Sanofi, which is practically wiping out your whole immune system. We all know that your immune system for infections, for your general immune surveillance is quite important. So another sweet spot of the program is that we leave the immune system intact. We go specifically after the chronically LAG-3 activated T cells. We leave the B cells alone, the naive T cell repertoire we leave alone. That's all nice and fine. So it is a very targeted approach.
And how important are those LAG-3 activated T cells? Is that a major driver of these diseases? Or is it just like a part of it?
Yes, that seems to be the case. Because they accumulate at the disease site, and LAG-3 we use simply as a marker, because you look there at the so-called exhausted T cells, those who constantly provide fire. And if the T cell does that, then they express LAG-3.
Right. Okay. Is there any other programs we should touch on?
I believe, we can talk for hours, but I believe these are the main programs. For me, it's important that it's understood that we are not a one-product company. We have also a small molecule. You mentioned that, an early-stage small molecule program. We have another program in autoimmune diseases. So we have 5 programs in total, 3 in oncology, 2 in autoimmune diseases, 1 in oncology actually out-licensed to Novartis. And we work in partnership settings. We follow the data, so that's our philosophy. Key value driver for the company moving forward is clearly the phase 3 in first-line non-small cell lung cancer, but there are other very important value inflection points in the short, mid, and long term.
Got it. So I guess to wrap things up, why don't we talk about your cash balance and what your investment priorities are? As CEO, you're the one that's going to have to make these calls. How are you thinking about that and your near-term priorities, long-term priorities, fundraising?
Yeah, thank you. This is actually a very important area. If you don't have the finance side and the budget under control, you run a big risk. So as important as it is to run clinical trials, to ensure operational excellency, regulatory affairs, the financial side of things is extremely important. So we have a very strong balance sheet of, according to our last 4C, more than 170 million Australian dollars cash on bank. So that's, I believe, very strong.
The market cap is about 140, just to give some...
Market cap is around 400 million.
Right, so 170 mil cash, 400 mil market cap.
Yes.
Understood.
So admittedly, a lot of room for improvement in terms of our share price, I believe, if you look at the intrinsic value. And if you do the net present value calculation, so I believe that we have quite a lot of improvement there, or chance for improvement. But we have a cash life, a so-called cash life. Biotech is typically not a revenue-generating business, so you invest heavily into R&D of more than 2 years, which is very good by the standards of our industry. We are supported by a number of very good institutional investors. We are dually listed, so we are listed on the Nasdaq. We are, of course, listed on the ASX. And you asked about priorities. First priority and key expense driver, if you like to look at it from that perspective, is clearly the registration trial in lung cancer, 750 patients, and then our other clinical trials. Compared to that, IMP761 is a smaller part of the budget. And you need to move your drug development forward holistically. So clinical data, as exciting as it is, it's not enough. There's manufacturing. So you need to show to the regulators that you can manufacture your drug on a reliable basis. There's a lot of work to do, and it's quite costly, of course. Regulatory affairs, quality assurance, preclinical side of things. So if you want to achieve a new standard of care, many things to work on, many expenses, and we negotiate every single agreement. So we have no shareholders' money, and the money the investors entrusted us with to lose. So it's our duty to have a lot of scrutiny on the budget, and I review every invoice just as a fun fact, because it's important that the team knows that every invoice will be reviewed, there will be questions being asked. We try to push on the cost without a compromise on quality, and you can save a lot of money with that.
Right. Have you found the dual listing?
On the one hand side, it's a very good thing because you have access to 2 different markets. It certainly increases your visibility in the United States for the pharmaceutical industry, for instance, so a variety of different investors. On the other hand side, you have, besides the ASIC, also the SEC. We need to comply with SOX, Sarbanes-Oxley Act, meaning you have to work under much more scrutiny than a normal listed ASX company. Because there you have to document your controls. These will be audited. So it's quite intense. And from a compliance and governance point of view, a very, very high standard. Could give a lot of confidence maybe to shareholders and investors. It's a lot of work internally. But there are sometimes good times because the Nasdaq is often driving the share price. We saw remarkable trading days in the United States where we have been the number 1 or number 2 traded stock on Nasdaq based on volume before Tesla and some others, where we had more than $1 billion in our stock on a single day. But it could also be, for instance, that political uncertainty in the States reflects into Australia. So there are pros and cons.
What are the thoughts on the incoming administration? Well, how do you think it's going to affect the FDA, drug development?
Yeah, that's a currently ongoing debate. I believe specifically the vaccine companies have been seeing a big hit as Mr. Kennedy, the incoming Minister of Health, doesn't seem to be a big fan in terms of, for instance, COVID vaccines. I don't go into specifics. There's also the debate regarding the FDA, but I believe the thought behind is to a certain degree deregulate, so have the business flourish. At the end of the day, looking at Immutep, if we deliver for the most important cancer indication, also in the United States, a clear improvement of the standard of care, this will be seen favorably by every administration. So we have to deal with the facts, how the facts are being presented. I believe the administration in principle is in favor of business. That's good. We take that and we have to do our job in terms of delivering and executing, and any uncertainty, which will certainly be there for the next few months, we simply have to deal with.
Okay, great. Marc, why don't we wrap up there? That was fascinating. Thanks so much for coming on.
Oh, thank you so much. Thank you for having me.
And I look forward to seeing those readouts later in the year.
Might be a Christmas present. Who knows?
I hope so. Thank you.